In this short video, we will try to find the answers of some common questions about rt-PCR. So the first question comes, what is PCR? A PCR is a procedure used to amplify or reproduce the selected section of DNA. That means the PCR targets to amplify some genes. And what we understand about rt-PCR, this rt-PCR is now being used worldwide to detect the COVID-19 disease. So we need to know what is rt-PCR stands for. This RT stands for reverse transcriptase PCR. As the PCR cannot amplify the RNA directly, the RNA need to be converted into a complementary DNA, and the reverse transcriptase enzyme is used first to convert the RNA into a complementary DNA. Then the PCR procedure will amplify the DNA, and the DNA detection will confirm the presence of RNA. That's why this procedure is called the reverse transcriptase PCR. But here it is a real-time PCR also, because we can see the progress of amplification or the number of copies of DNA produced in real time in a monitor. So it can be confusing that what should be the proper name. So it is a reverse transcriptase real-time PCR or rt-qPCR. Here Q stands for quantitative PCR. That means the amount of DNA that is produced can be seen in a monitor in real time. So after starting the procedure, we can see in the monitor the amount of DNA amplified by the graph. Here there are multiple graph represents the different samples, and the progression of the curve from the baseline indicates the amplification process, or this is the positive results where there is the presence of RNA in the sample. So does a rt-qPCR method needs a specific type of thermal cyclers, or a conventional cycler is enough? The answer is yes, that the rt-qPCR needs a special type of thermal cycler that need to be presence of optics and light sources. The light sources is laser or LED type of light through which the machine can detect the amount of DNA produced in real time, and that can be converted into a graph. But in conventional or endpoint PCR machines, they do not have this light system or optic system. That's why the products of PCR cannot be recognized in the machine by itself. So after the completion of PCR, the product of PCR need to be visualized by different staining methods and electrophoresis. So that is the difference between the thermal cyclers of conventional endpoint PCR and the rt-qPCR cyclers. What is the maximum patient sample can be tested at once with a 96 well PCR machine? That means how many samples can be run at one time in a PCR machine which have 96 wells. So in 96 wells, out of 96 wells, one well will be used for a negative control. That is just a water sample, but that will be processed with other samples, and another well will contain a positive control, which is a confirmed SARS coronavirus 2 nucleic acid will be present there. That is a positive control. So 2 well is used for this control, and the rest 94 wells can be used for 94 patient sample at once in a 96 well PCR machine. So we can see that maximum 94 patient sample can be run, but for the more clarification and for more control can be run for extra confidence, so that the patient sample that can be compared with the positive and negative control, and the result will come accordingly. Next, how long does it take for rt-qPCR to complete one cycle? That means from the beginning of the sample processing and up to the result. So broadly, it takes for RNA extraction and other procedure, it may take 1 hour, and after the RNA extraction, the PCR stays, and in the thermal cycler it will take 4 hours to complete the whole thermal cycling and the DNA amplification, and after that the result will be coming. There are automated extractor machine available nowadays, and they are used mainly in the big laboratories where the patient sample load is very high, and also depending on the income of the lab. So if we use the automated sample processing units or automated RNA extractor machines, the time will be a bit less from the conventional method. So how to interpret the results of rt-PCR machine, or how we will read the result from the rt-PCR machine. The rt-PCR machine gives curve or generate curve in the monitor from the samples. So the curve should be read, and the curve that is generated by the sample, if it is not a flat line, then it is positive. The samples which are negative, they give a flat line, and the samples that are positive, that means the samples containing the RNA of that particular virus, it will be amplified and the curve will go up. So this curve that is not flat and goes upward is positive. So bigger concern is the false negative, and it has many reasons. And false positive result is unlikely because the RNA will be amplified when it is present. So rigorous use of control, both positive and negative control, is mandatory, and it should be compared with the sample result. False positive occurs in very few cases, and this is mainly due to the contamination of the sample from a positive sample or mixing up, and that is very less in number. Next question is how to validate our test procedure. That means the procedure we are using, and that is correct and giving the highly sensitive result, we need to validate that. So the sensitivity of the test depends on many factors. One may be the extraction method, then the kit we use, the manufacturer of the kit, and also the gene, our the primer targets. So these are the many variables on which the sensitivity depends. So we have to validate our test procedure by using rigorous using of standard control, and comparing our test with the standard control will give the validity of the test, and this also indicates the sensitivity of the test procedure.